Hello student, welcome in Department of Nanoscience and Nanotechnology S.Y.B.Sc class. In last lectures, we have been seen the mechanical and magnetic properties of materials. In today's lecture, we will see the optical properties of materials. The optical properties of material define how it interacts with light. So, first optical property is luminescence. Luminescence is emission of light by a substance not resulting from heat. It is thus a form of cold body radiation. It can be caused by chemical reactions, electrical energy, subatomic motions, or stress on a crystal, which all are ultimately caused by spontaneous emission. It is in contrast to light emitted from incandescent bodies such as burning wood or coal, molten iron, and wire heated by an electric current. It may be seen in neon and fluorescent lamps, television, radar, and X-ray fluoroscope screens. In all these phenomena, light emission does not result from the material being above room temperature, and so luminescence is often called cold light. Luminescence emission occurs after an appropriate material has absorbed energy from a source such as ultraviolet or X-ray radiation, electron beam, chemical reactions, and so on. The energy lifts the atom of the material into an excited state, and then because excited states are unstable, the material undergo another transition back to its unexcited ground state, and the absorbed energy is liberated in the form of either light or heat or both. Types of luminescence are photoluminescence, chemiluminescence, crystalloluminescence, electroluminescence, mechanoluminescence, radioluminescence, and thermoluminescence. Next is phosphorescence and fluorescence. With respect to organic molecules, the term phosphorescence means light emission caused by electronic transitions between levels of different multiplicity, whereas the term fluorescence is used for light emission connected with electronic transitions between levels of like multiplicity. Fluorescence is an almost instantaneous effect ending within about 10 raised to minus 8 seconds after excitation. So next is photoluminescence, which is also known as spectrofluorometer. It is one of the type of luminescence. Photoluminescence or PL is light emission from any form of matter after the absorption of photons, that is electromagnetic radiation. It is one of many forms of luminescence and is initiated by photoexcitation, that is excitation by photons. Photoluminescence processes can be classified by various parameters such as the energy of the exciting photon with respect to the emission. Resonant excitation describes a situation in which photons of a particular wavelength are absorbed and equivalent photons are very rapidly re-emitted. This is often referred to as resonance fluorescence. Photoluminescence is an important technique for measuring the purity and crystalline quality of semiconductors and for quantification of the amount of disorder present in a system. Photoluminescence excitation is a specific type of photoluminescence and concerns the interaction between electromagnetic radiation and matter. Next is basic principle of photoluminescence. Photoluminescence spectroscopy is a contactless, versatile, non-destructive, powerful optical method of probing the electronic structure of materials. Light is directed onto a sample where it is absorbed and imparts excess energy into the material in a process called photoexcitation. One way this excess energy can be dissipated by the sample is through the emission of light or luminescence. In the case of photoexcitation, this luminescence is called photoluminescence. PL spectrum is quite different from absorption spectrum in the sense that absorption spectrum measures transitions from the ground state to excited state, while photoluminescence deals with transitions from the excited state to the ground state. When energy is absorbed by electron, it will jump from ground state to excited state, and as we know, the excited state is unstable state, so electron will go from excited state to ground state and it emits the photon, so this photon is known as luminescence photon. Next is experimental setup. A spectrofluorometer is an analytical instrument used to measure and record the fluorescence of the sample. While recording the fluorescence, the excitation, emission or both wavelength may be scanned with additional parameters. Variation of signal with time, temperature, concentration, polarization or other variables may be monitored. Figure shows the block diagram of fluorescence spectrometer. Fluorescence spectrometer use laser source which contains wavelength selectors, sample illumination, detectors and corrected spectra. So this is the block diagram of PL spectrometer or spectrofluorometer. This is source, this is excitation monochromator, this is sample cell, these both are the slits, this is emission monochromator. The basic function of monochromator is to pass the single wavelength. This is detector, amplifier and record, which records the variation of signal, time, temperature, concentration, polarization or other variables which may required. We will see the description of each and every block. First of all, source. The continuous light source is 150 watt ozone free xenon arc lamp. Light from the lamp is collected by a diamond turned elliptical mirror and then focus on the entrance slit of the excitation monochromator. The lamp housing is separated from the excitation monochromator by a quartz window. Next is monochromators. It contains two monochromator, excitation monochromator and emission monochromator. They use all reflective objects to maintain high resolution over the entire spectral range and minimize rediffraction. Next is gratings. The essential part of a monochromator is a reflection grating. A grating disperses the incident light by means of its vertical lines. A spectrum is obtained by rotating the gratings contain 1200 lines, that is grooves, per mm. Next is slits. The entrance and exit ports of each monochromator have continuously adjustable slits. The emission monochromator's slit control the intensity of the fluorescence signal recorder by the signal detector. The wider the slits are, the more light falls on the sample and detector, but the resolution decreases. Next is shutters. An excitation shutter is located just after the excitation monochromator's exit slit. The shutter protects sample from photodegradation from prolonged exposure to the light source. An emission shutter is placed just before the emission monochromator's entrance and protects the detector from bright light. Next is detectors. It contains two detectors, signal detector and reference detector. The signal detector is a photon counting detector. This detector is photomultiplier tube which send the signal to a photon counting module. The reference detector monitors are the xenon lamp in order to correct for wavelength and time dependent output of the lamp. This detector is a UV enhanced silicon photodiode which is just before the sample compartment. So this is the PL spectroscopy or spectrofluorometer. Next is electroluminescence. The mechanism behind electroluminescence is the radiative recombination or spontaneous emission. When a semiconductor is subjected to an electric current, recombination of the electron-hole pairs takes place. These excited electrons release energy in the form of photons, resulting in the emission of light. Electroluminescence is the phenomena where a material emits light when electricity is passed through it. Electroluminescence generally involves a material producing light without producing heat. Electroluminescence is produced when an electric current is passed through a semiconductor with tiny holes. As excited electrons pass over these holes, they are emitted into the free air as particles known as photons. The diagram shows a very simple representation of two electrodes and a thin layer of electroluminescent material sandwiched between. Each electrode is a layer of conducting material. It is so thin that light is able to pass through and be emitted to the surroundings. Electroluminescent material will only give off light when an alternating voltage is supplied to it. It does not work on DC. Next is applications. Electroluminescence has many uses. It is used in automobile dashboards to illuminate the car's meters. Likewise, it is used in electronics to illuminate small LCD panels. Also, any billboard that is illuminated at night is generally using electroluminescence. Electroluminescence should not be confused with neon and fluorescent lamps. The first thing to understand is how a circuit sees a piece of electroluminescent material. It sees it is a capacitor. The resistance between the two electrodes is almost infinite and thus DC will not pass through it. But when an alternating voltage is applied, the buildup of a charge on the two surfaces effectively produces an increasing field called electric field, and this causes the phosphorus within the field to emit light. The increase in voltage in one direction increases the field and this causes the current to flow. The net result is the current flows into the electroluminescent panel and thus energy is delivered to the panel. This energy is converted to light via the panel, and this conversion is a very efficient process since no heat is produced in the process. This makes EL a very efficient form of illumination. Electroluminescent wire or cable is constructed with a thick stuff inner wire surrounded by a coating of light emitting phosphorus, and around this is wrapped a very fine outer wire. An outer clear plastic jacket or sheath protects the chemicals and insulates the voltages on the wire from external leakage. Thank you very much.